Yes, I have the pleasure to introduce Dan Kastner here. But first I would like to welcome Crafoord Laureate, the presidents of the Swedish Academy of Science and the Crafoord Foundation, and the ladies and gentlemen. So you're all welcome here. I will give a background of the area and the prize that Dan Kastner has won. And I think that Dan, as all of us, has a dream to really understand the causes of diseases. And there are basically only two factors that we need to understand. It should be quite simple. We need to find the genes that cause diseases, and we need to find the environmental factors. But we have not succeeded. There are problems. And these problems are that we have many genes. In fact, we are 3 million base pair nucleotides that built up our genetic code. And we also have an unlimited and uncontrollable environmental factors, whatever that can be. And these all interact with each other and cause a disease. It's really, really very complex. And that's the thing we don't understand. These diseases that is caused by all these interactions with all the genes together, we call complex diseases. It's not one gene that causes it, it's many genes that cause it, many environmental factors. And this causes diseases like rheumatism, cancer, atherosclerosis, dementia, infections, even traffic accidents, if you think of it. So, as a scientist, we have to think how to really find these genes. And there's been a lot of investments, and one big hope has been for many years, we sequence the human genomes, then we know who we are and which diseases we have and will get. And so we put $3 billion into this project. It's done. Actually, the final only left of 0.5% left of the DNA sequence is just published. And today you can sequence for $500 yourself. But we didn't really find the genes for complex diseases. They are there in front of us, but we don't understand which they are and how they work. So what you do is that you have the sequence of the genome, you compare that with all the disease traits that clinicians has described, and then you do association studies. You compare the data and hope to find some link in between with the computer. And we found a lot of what we call loci, parts in the chromosomes that correlates with disease. We call that associations. But very few, if any, real genes with nucleotides has been really found, actually, to be honest. And we don't really know of what these loci really do for disease. So the clue to complex diseases has been very difficult to find. This is just one example. I have a picture here of this lady. You see the erythema or the inflammation on the neck. She has systemic lupus erythematosus. Lupus is wolf, so it's just characterized by a bite of a wolf over the neck. It's systemic. That's red skin, a lupus bite. It's a terrible disease. It's common. It's complex. It affects young people and women. And we don't know the cause. We don't know the genes. And the genome factors, we know a lot of these loci, chromosomal fragments. Actually, these are the loci that we have here. And it's gene names here. But we can't believe that because this is just an example of where they are in the fragments in here. They indicate different mechanisms how it works. There's one gene that we actually know of, and interestingly, this gene is is not present, has not yet been sequenced. It's one of the dark spots that we don't know of. So how could you figure out? That was figured out from animal models. And going back to humans, and this is the most important gene for this disease. So there is some light in the tunnel, and it's possible to go further in this. But there's an alternative strategy also. And Don Kastner, I think, has done a great work in enlightening this alternative strategy. And there's been a lot of success using the human genome to figure out what we call monogenic diseases. That means that a gene that could cause something directly without a big interaction effect of environmental factors or other genes. And then you can explain the relation between them and then go further to understand how they work and pathways. And Don actually found with his work on this a new class of diseases that he called autoinflammatory diseases, which is part of the immune system attacking the body, giving different kind of symptoms. It's a big, diverse group of diseases, but recurrent fever is one of these groups. And so they could clone and identify different genes in the chromosome. This is chromosomes here, and this is the different places of different genes that actually call these autoinflammatory diseases. And I think that's been a fantastic discovery to clinical medicine now. We can explain new diseases. We can explain, which is really very important, the molecular basis, how they are caused, and we can now go further into this. This makes us learning a lot about not only clinical science and theory, but also biology, because this could deepen our the mechanism, understanding the mechanism how these diseases goes about, and it also could give light to complex diseases, because these genes could also contribute these pathways to these bigger diseases as well. Yeah. So with this word, I would like to invite Don up here, so he can explain in a more clear way what this is about, what autoinflammatory diseases are. We are so happy here to have you here as a prize winner.
Well, thank you so much, Professor Holmdahl, for that really kind introduction. And it's just absolutely wonderful to be here today to talk to you about a subject that's near and dear to my heart, and that is the systemic autoinflammatory diseases. And before I get started, of course, I'd like to thank the Crafoord Foundation. I'd like to thank the estate of Holger and Anna Greta Crafoord for setting up this prize, and of course the Royal Swedish Academy for choosing this work for this incredible honor. So in any case, what we are going to be talking about today is cutting the Gordian knots of inflammation with the shears of genomics. And for those of you who are not students of antiquity and therefore don't know anything about the Gordian knot, perhaps we should start there, just by saying that the Gordian knot was this legendary knot that apparently was in some village in Asia Minor. And supposedly it was a very difficult knot to untie. And whoever was able to untie this knot would become the ruler of the world. So anyway, Alexander the Great happened through this village. He came in and sized up the knot, determined that it would, in fact, be very difficult to untie that knot. And so he pulled out his sword and cut the knot. And so the idea of the Gordian knot is basically something that's a very difficult problem to solve, and cutting the Gordian knot is using some unconventional approach to do that. Now, at the time that I got involved in this work, the field of genetics, and at least the field of genomics, was just in its infancy. The Human Genome Project hadn't even started at that point. I basically was becoming interested in this in the mid-1980s. The Genome project didn't start until 1990. So definitely this tool, this sword of cutting the Gordian knot, was something that developed over the course of time. It was not a ready sword for doing what we needed to do. But it was a lot of fun, and I hope that you'll have some fun as we talk about this over the course of the next little bit. So my first exposure, actually, to this sword of genomics was when I was 12 years old. And at that time, my family subscribed to that esteemed medical journal, Life magazine. And Life magazine had a cover story on solving the genetic code. And this was not the Watson-Crick structure of DNA discovery. Instead, it was Marshall Nirenberg's discovery of the code of DNA that then could be translated into the sequence of proteins that are encoded by that DNA. Well, that prompted me to do a 7th grade science fair project on DNA with pipe cleaners and jelly beans, but that was about all that happened from that for a while. Fast forward a little bit, and here I am finishing the PhD component of the MD-PhD program at Baylor Medical School. I was doing that in mouse immunogenetics, and yes, you can see here on my shoulders, those are live mice crawling around. And so I had, at least at that point, developed some interest in immunogenetics. And a little while further on, I finished my internal medicine residency. And a few days after finishing my internal medicine residency, I married Margaret Beckwith, who has been my collaborator in, at least in terms of advising me on things going forward over the course of the last, whatever it is now, a few years, since 1985. So in any case, and we were about to set out to do postdoctoral fellowships at the NIH. Now, at that time, not only did I have, of course, the new adventure of being married to Margaret, but also I had the goal of marrying my basic science activities with my clinical activities. That was really the dream, was to figure out a way of having some disease or family of diseases that I would be studying in the laboratory that would in some way have an impact on the treatment of patients and understanding their diseases and understanding something about biology. So what better place to do that than the NIH, the National Institutes of Health, and this is the clinical center of the NIH. And it had the advantage, still does, of being the largest hospital in the world that's dedicated to research on patients. Every patient that's admitted there is a research subject. This is what it was back in 1985 when I first went there. But the other thing is that it had a culture, and still does. And that culture is a culture of really taking on long-term, high-risk, high-reward kinds of projects. And so that's what really I wanted to do, and this place really made that possible. So, in any case, I got to the NIH, and within the first few months that I was there, actually, I saw a patient. And this was a young man of Armenian ancestry who we saw in the arthritis clinic. And this man had a history of recurrent episodes of fever, usually occurring on the order of once a month or so. And with that fever, he would have severe arthritis, affecting usually one or sometimes a couple of joints at a time. And oftentimes it was a knee or an ankle, and during that time he'd have massive swelling of the joint. He would have incredible pain in the joint and would be hobbled for some period of time, for a few days, and then between the episodes he was totally fine. So what did he have? Well, he had a condition called familial Mediterranean fever. And we deduced that at least in part because of the clinical story and in part because of his Armenian ancestry. So FMF is a disease that by then was known to be a recessively inherited disease seen in certain ethnic groups, in Armenians, Arabs, Turks, Jewish people, and people of Italian ancestry. And so, just to illustrate, these are just indicating when it was that I saw this patient. So the clinical features of FMF are illustrated here. The patients have fever. They can have sharp abdominal pain, sometimes to the point, actually, where they'll have an appendectomy because they're suspected of having severe appendicitis. They have sterile inflammation of the lining of the abdominal cavity. They have pleurisy, sharp stabbing pains in the chest caused by inflammation around the lungs. They also have the arthritis, as I described before, and they can develop amyloidosis, which is the deposition of a cleavage product of one of the acute phase 1 reactions, one of the substances that's made during the inflammatory episodes, and it can deposit in the kidneys and other vital organs and cause renal failure and even death. So at that point, this was a disease that was interesting. It was a disease that was not understood at that point. And because of the fact that it was known to be a recessively inherited disease, it was then a disease where possibly we could use then the sword of genomics to cut through the Gordian knot. And so that's basically what we tried to do. So I established a collaboration with this guy here, Mordechai Pras, and he, at that time, he's still around and still one of the world's clinical leaders in the study of familial Mediterranean fever. Here he's shown at the Dead Sea with a patient with psoriatic arthritis getting a mud treatment. This was back in the summer of 1989 when I first went to Israel. And we both saw patients in his clinic and also went to people's houses. And this slide is just a picture of one of the families that we drew blood on. Their faces have been obscured just for confidentiality reasons. But in any case, over the course of 6 weeks, we drew blood on 62 families and made 347 cell lines from individuals from those families. Well, in any case, from that, then that gave us at least the beginning materials, the DNA materials that we would need in order to figure out the gene that is mutated in FMF. At that time, there were only a few markers in the human genome that were known. There were about 300 markers, and that's about the minimum that you would need in order to map a disease gene. But we were not deterred by that, and I set up my own lab. This is in the early 1990s. I was a tenure-track investigator. Ivona Aksentievich was my first postdoc, who is still a incredible collaborator and very productive person in the field of human genetics, and Louis Gruberg, who was a internal medicine resident from Israel. And with Elon Pras, who is the son of Mordechai Pras, who joined us a little bit later, we were able to map the gene for FMF to the short arm of chromosome 16, which did take a fair amount of effort in order to do that. But then we were faced with the problem of, once we had this region of chromosome 16, of actually figuring out what the gene was. And the problem was that the region on chromosome 16, we didn't know exactly how big it was. We knew that it was probably in the millions of base pairs long, but because the Genome Project had not yet been completed, we didn't have a sequence of that region. We didn't have really any clues. We had to generate them ourselves. And so we became the Genome Project for this region. And eventually, we identified a couple of markers, DNA markers. One of them from the polycystic kidney disease region of chromosome 16, and the other from what was called the Rubinstein-Tabi region of chromosome 16. We knew those were the boundaries. We didn't know how far apart they were or exactly what their nature was. And so from that, we were then able to pull out chunks of DNA from chromosome 16 to be able eventually to assemble a map of that part of chromosome 16. And so essentially, we pulled out chunk after chunk, starting on one end and moving in. This was the telomeric end on that end, and then also coming from the other end. And eventually the two maps met. Now they didn't necessarily have to meet. We didn't know for sure if the library of chunks was actually going to be complete, but it turned out that it was. And this then allowed us to develop other markers that would allow us to narrow the region, so that we found that those original two markers were about 1 million base pairs apart from one another. With additional markers that we identified, we could then narrow things down to around 100,000 base pairs. And then we had to use a special kind of vector that would allow us to pull out chunks of coded genes, and from that we had one chunk that allowed us then to pull out actually the gene that is mutated in FMF. So altogether, it took us from the summer of 1989 until the summer of 1997, so about 8 years to find the gene using these approaches. Well, in any case, it turned out to be really great in the sense that it was exactly what we wanted. A new gene that nobody knew anything about before. So it really was then a way of beginning to understand the control of inflammation. But the problem was that it was exactly that, a gene that nobody knew anything about. And so it wasn't like it was immediately clear exactly what one could learn from this gene. Well, one of the things that we learned relatively soon from this gene is that it has this domain at its N-terminus that is called the pyrin domain. We named the protein that's encoded by this gene pyrin after pyrexia for fever. So in any case, this pyrin domain, it turns out, is the prototype for a domain that is found in some 20 different proteins that are involved in regulating human inflammation. And essentially, this domain forms 6 alpha helices, which allow for the formation of a charged dipole with positive charges in blue and negative charges in red. And so basically you can get a interaction between the pyrin domain of one protein and the pyrin domain of another protein. And this is actually a basic way that proteins in the innate immune system can interact with one another. And as I say, there are 20 proteins in the human genome that have this pyrin domain, and then there are actually 3 other domains that are similar to pyrin domains that also interact in that way. Well, while we were beginning to understand a little bit about that, Mike McDermott, a former fellow of mine, and Ivona Aksentievich joined forces to start looking at patients with recurrent fever syndromes that weren't FMF. So we started seeing just patients that were referred to us who had unexplained fevers, but we knew that they didn't have familial Mediterranean fever. And one group of these patients had prolonged episodes of fever. They were not of Mediterranean ancestry, at least some of them were of Irish ancestry, and were therefore called familial Hibernian fever for Irish fever. And they didn't respond to the usual treatments for FMF. And within a relatively short period of time, Ivona and Mike had discovered that those patients actually have mutations in the receptor for tumor necrosis factor, which was at that point already a known mediator of inflammation. And so they called this disease TRAPS for the TNF receptor-associated periodic syndrome. And because we had by then, over the course of a couple of years, between 1997 and 1999, discovered a couple of diseases in which patients had these unexplained recurrent fevers, but we now had an explanation in terms of a gene, without the autoantibodies that one would see in autoimmune diseases. We thought, well, maybe it's a new family of diseases. And so we hypothesized that we had a new group of diseases which we called autoinflammatory diseases. And a year later, we published a paper in which we speculated on all of the diseases that we could think of that maybe were also autoinflammatory diseases. And we had quite a long list of these diseases. And a couple of the diseases that we talked about in that paper were these two, familial cold urticaria and Muckle-Wells syndrome. And we were just hypothesizing, based on what we knew of the clinical features of these diseases, that maybe they too were autoinflammatory. And then, much to our delight, about a year later, Hal Hoffman at UCSD published a paper in which he showed that those two diseases that we said were probably autoinflammatory, in fact, were caused by mutations in a gene that has a pyrin domain. Remember, that's the domain that basically is on the N-terminus of the pyrin protein and that allows for protein-protein interactions. So we were right, at least for those diseases, that probably they were autoinflammatory. And within a fairly short period of time then, it was recognized that that gene encodes a protein that regulates interleukin-1, which is a mediator of inflammation. And Phil Hawkins and Helen Lockman and Mike McDermott did a study showing that, in fact, an inhibitor of IL-1 would be effective in treating those patients. And then our group discovered that there was an even more severe disease called NOMID, neonatal onset multisystem inflammatory disease, that's also caused by mutations in the same gene that also causes Muckle-Wells syndrome and familial cold urticaria. And so in that condition, in that disease, patients have not only a hives-like rash and conjunctivitis that you can see in these other two conditions, but they also get aseptic meningitis, inflammation of the lining around the brain, which can lead to blindness and deafness and intellectual disability. And shortly after we had found mutations in this gene in these patients, we had accumulated a cohort of about 18 patients with this condition. And so we decided that really it was the only responsible thing to do to try to treat those patients with anakinra, the IL-1 receptor antagonist. And it was really, really an incredible thing. So what happened was that we started these patients on anakinra, and within days their rash would go away and their conjunctivitis would go away. But within 2 or 3 months, the aseptic meningitis went away, and the cochleitis, the inflammation of the cochlea, went away. And basically, at this point, these patients we started on treatment with anakinra back in the early 2000s, and these kids are now graduating from college. I mean, if you wanted to believe in molecular medicine, this really was a case where it worked. So in any event, this really sort of solidified this idea of autoinflammatory disease, where we had found a couple of disease genes that gave us the thought that perhaps this was a type of human illness. We had predicted that then these patients would have certain biologic connections in terms of the cause, and one could actually predict treatments based on those insights, treatments that work. And so that not only helps the patients, but actually solidifies some of the understanding that we have in terms of these diseases. And at this point, we now recognize that there are sort of two flavors of immunity. Adaptive immunity, in which T and B lymphocytes are the major players. The receptors somatically rearrange and mutate. And the autoimmune diseases are characterized by high titer autoantibodies or antigen-specific T cells. With innate immunity, it's a different cell type that's involved. It's myeloid cells, granulocytes and monocytes. The receptors are hardwired in the germline genome. And these autoinflammatory diseases are characterized by seemingly unprovoked inflammation, but without the autoantibodies or antigen-specific T cells. Well, what happened then at that point is that there was a revolution in human genomics. And that revolution was the next-gen sequencing revolution. And so basically, as you can see on this graph, the cost of DNA sequencing and the cost of computing went down more or less in parallel up until around 2007 or 2008. And then at that point, the cost of sequencing came way down relative to the cost of high-performance computing. And that really revolutionized things and has allowed advances that we would not have dreamt of before. So I'll just give you one example of that. And this is the work of Qing Zhao, who was a fellow in my lab at the time. And so we were referred a patient, a little girl from New Jersey, who was 2 years old at the time. And she had had recurrent fevers over her first 2 years of life. And then she had a catastrophic neurologic event. And she was referred to us because her physicians thought that perhaps she had this disease, NOMID, that I told you about. Well, we saw her at the clinical center, at the NIH Clinical Center. She didn't have NOMID. She didn't have the skin rash of NOMID. She didn't have the aseptic meningitis by MRI scanning. She didn't have a mutation in the gene that's mutated in NOMID. She did have multiple strokes in the deep brain nuclei, and she had some sort of an inflammatory disease that we didn't know what it was, and so we just treated her empirically. And then, about 10 years later, we were referred a little girl from Texas. She was 6 years old at the time that we referred her, and she had had 6 strokes over the course of her 6 years of life. So here we had these two patients, one from New Jersey, the other from Texas, both of whom had recurrent fevers and strokes. And so we thought, well, maybe they had the same disease. Maybe it was the same gene that was involved. And so we used actually then whole exome sequencing of all of the genes in the genome to try to see whether or not they had mutations in the same gene. And lo and behold, they did. And they turned out to have mutations in a gene that's called ADA2, adenosine deaminase type 2. And we published this in 2014. This is just some images from that paper. These arrows point to the strokes that some of these kids had. They also had this blotchy-like rash shown here. They also could get inflammation of the blood vessels, and that could cause, in some cases, necrotic lesions, say, on the ear. It's a form of vasculitis. But then we were faced with another problem. And that problem was that now that we knew what this was, people started referring patients to us that had kids that had recurrent fevers and had strokes. And the problem was that these kids didn't just have one stroke. They would have recurrent strokes, like 1 every 3 years or so on average. And they had been on all sorts of different treatments, and nothing had been very effective in controlling the strokes. So here we had all these kids that we were following that had strokes already and that we knew were going to have more strokes, and what to do. So at that point, really one of the only kinds of treatments that hadn't been tried very much in these patients was inhibitors of tumor necrosis factor. And so Mandy Umbrello in my group started a study looking at TNF inhibitors in these kids. And we looked at 15 children with this condition, and so at least initially, you can see that the inflammatory markers came down. And before going on TNF inhibitors, these 15 kids had had 55 strokes over the course of about 2,000 patient months of observation. And then after going on TNF inhibitors, we had about 700 patient months of observation, and the number of strokes was 0. So terribly effective. It's not a targeted treatment in the sense that we didn't do this because we knew that this had something to do directly with TNF, but because of the fact that we were able to group these patients together in some sort of a categorization where we could actually study them as a group, we were able to figure this out. And now kids that have this disease, of course, are treated with TNF inhibitors as a routine thing. Well, I can tell that I'm probably going to at least reach my time, my end time. So towards the end of this talk, I'd like to talk about 3 riddles of the modern day autoinflammatory sphinx. So the first riddle that we will talk about, and I'll try to talk fast here. Riddle number 1: why are there such high carrier frequencies for familial Mediterranean fever in Middle Eastern populations? And so you might say, well, I didn't know there were. Well, there are, and in fact, if you look at 4 ethnic groups that have a high frequency of FMF, you can see that the total carrier frequency for FMF in these populations is somewhere between 10 and 12%. To put that into some kind of perspective, the carrier frequency for cystic fibrosis, which is the most common lethal recessive disorder in North America, is about 4%. So a 10 to 12% carrier frequency is really, really high and makes you think that maybe something was selecting for that over the course of history. To actually establish that there might be some sort of selection, you have to do some population genetic studies, and at least some of that is illustrated on the upper left-hand corner of this slide. So what we're looking at here is in the Turkish population, which is one of the populations where FMF is common. We're looking at the size of the DNA fingerprint that's either associated with FMF mutations or with the wild type version of the FMF gene. And the width of either this peak here, which is for the wild type allele, or the blue peak, which is for the mutant allele. And you can see that this width is a lot greater than the width of this thing here. That's exactly what you would see with some sort of selection. And one can even then do calculations of what is the coefficient of selection for these FMF mutations in the Turkish population, and you come up with a number of 0.077 for this one particular mutation. That compares with a selection coefficient of 0.056 for lactase persistence in Northern Europeans. That's the ability to digest cow's milk as an adult. And that we know has been under selection in the Northern European population. So this is telling us that, in fact, there was selection for FMF mutations in these populations. And one can even, using some mathematical approaches, you can estimate when these FMF mutations arose. And it's somewhere between 2,000 and 5,000 years ago. So then you can think, well, what would have been selecting for FMF mutations in the Middle East? So one of the things that could have been selecting for it is the plague, Yersinia pestis. And so if one thinks about infection with Yersinia pestis, it turns out that actually pyrin, the FMF protein, is an important defense against plague because it leads to the production of interleukin-1, which is a cytokine that can counter plague. Now, plague happens to have a toxin called YAP-M that can inactivate pyrin in normal people. But actually in FMF patients, they have a variant form of pyrin that is not susceptible to that inhibition. And so for patients with FMF, which are less than 1% of the population, you don't have that blocking effect of YAP-M. But what's more is that for FMF carriers, which is a much larger percentage of the population, for a recessive disease, the carrier frequency is a lot higher than the affected frequency, that there also you have a resistance to this YAP-M protein. So basically, the Faustian bargain is that you have protection against plague in 10% of the population or something like that, and the price that you pay is that some small percentage, less than 1%, die of this genetic disease. So that may well be what's going on. And one can simulate what are the carrier frequencies then for FMF in the Turkish population, and you can see that the numbers that one comes up with are actually quite close to what we observe in the modern day Turkish population. Okay, the second riddle. This riddle, you got to hear this riddle. Then maybe I won't tell you about the third riddle. You know, that can be for the next time. So the second riddle: how do old people get autoinflammatory disease? That's a good one for people like me, anyway. So in any case, the approach that we usually use for finding disease genes is that we start with a whole bunch of people, and then we try to sort out a few people that have some phenotype in common, some clinical characteristics in common, you know, a certain kind of arthritis or a certain kind of skin rash or whatever. And then we try to find a gene that's in common amongst those people. And that's what we usually do. Now, some of the people in my lab, David Beck was one of them, decided that we would try the opposite approach, genotype first. And so there we're looking at particular genes, and then we're trying to project that onto the population to find people that have mutations in those genes. So it's just an opposite way of doing it. And in doing that, David found that there were mutations in this gene UBA1 that in fact are found in certain individuals. They're middle-aged or older men, and they happen to have mutations in the peripheral blood. They have two forms of the gene in the peripheral blood. But rather than having in all of the cells of the peripheral blood both forms of that gene, they have in one subpopulation of cells the mutant form, and in the other subpopulation of cells, the wild type. And so you see that in myeloid cells, they have the mutant form, and in lymphocytes, they have the wild type form. And so essentially, it's somatic mutation. It's what happens to all of us as we get older, and it's one of the causes of cancer. And what David found was that, in fact, the people that have those mutations, in fact, carry a bunch of different diagnoses, such as relapsing polychondritis. That's where you get inflammation of the cartilage of your ears. Sweet syndrome, which is a disease where you get boils all over your body. Polyarthritis nodosa, giant cell arteritis, are conditions where you get inflammation of the blood vessels. But they all have mutations in this UBA1 gene. And so he came up with the name for this disease, VEXAS, which stands for vacuoles, because they have vacuoles in their bone marrow precursor cells. E1, ubiquitin activating enzyme, X-linked autoinflammatory somatic syndrome. And the clinical features are illustrated here. These patients can have inflammation in the lungs. They can have inflammation of the blood vessels, this inflammation of the ears, and these vacuoles in the precursor cells in the bone marrow. So the big picture from this little story is, number one, it's a paradigm shift in gene discovery using a genotype first rather than a phenotype first approach. approach. Secondly, adults develop autoinflammatory disease the same way that they can develop cancers, by somatic mutations. And thirdly, throw out the textbooks, because in fact, the textbooks, give you an organizational scheme that's not as good as the organizational scheme that you would have if you figure these things out by the genetics. So in any event, that's it for that. We will not do riddle number 3 because we lack the time to talk about riddle number 3, but I will just finish up with some take-home messages. The autoinflammatory diseases are disorders of innate immunity. The monogenic autoinflammatory diseases have taught us a great deal about how innate immunity works. In some cases, targeted therapies have been transformative while validating our understanding of the underlying disease. Pathogens sometimes select for common genetic variants that protect against the pathogen but predispose to autoinflammation. Somatic mutations account for some adult-onset autoinflammatory diseases. And then the last thing that I didn't show you the data for is that cassettes of relatively recent, relatively frequent genetic variants confer risk of common autoinflammatory conditions. And this is just a list of the different diseases that we have discovered over the course of the last few years since we started working on this. A couple of them, of course, were known diseases, familial Mediterranean fever and NOMID. There's 16 diseases on this list. 14 of them were not known back in 1985 when we first started doing this. And then some common diseases where we've also found susceptibility loci. Here's the people in the lab that contributed to this, and this slide just illustrates, this is before the pandemic, the people in the lab, the people in the clinic, and the fact that this is a collaboration between the two. We have a lot of other collaborators as well. These are the patients that obviously would make this all happen. These just happen to be patients with DADA 2. And my family, of course, my wife Margaret, my son Ben, and my son Nathan, all of whom put up with these shenanigans. And then finally, one last thing that I will say just a little bit about for half a minute is that I've tried to at least use some of the proceeds from the Crafoord Prize to recognize some of the people that contributed to my work. So we've set up an awards system for the International Society of Systemic Autoinflammatory Diseases. And so now going forward for the next few meetings of biannual meetings of that society, there will be a Basic Sciences Award named after the late Jörg Chopp, who discovered the inflammasome, a Human Genetics Award named after Isabelle Tuitu, who is another of the geneticists in the field, a Clinical Investigation Award named after Mordecai Pross, Therapeutic Trials Award named after Alberto Martini, and then a Trainee Award named after Charles Dinarello, who actually was a Crafoord Laureate back in 2009. And so the idea is that new investigators in the field will be recognized, and in so doing will honor these people as well. So anyway, I've overstayed my welcome, I can tell, but in any event, thank you all very much for your time, and I'll try to answer any questions that you may have.